Happy New Year. My name is Joe Komsky, and I'm the Senior Director of Philanthropy here at Sanford Burnham Prebys. Once again, the Earth has made its 365-day trip around the sun, and so our thoughts turn to renewal and change. Did you make New Year's resolutions this year? Did you resolve to start keeping a journal? Or maybe you resolved to give more money to charity? I can never keep my resolutions, so I've started resolving to do the opposite of what I want to do. So this year I'm going to try to exercise less and read fewer books. But by far, the most popular resolution that people make is to improve their eating habits and lose weight. And so today we're going to provide information about the importance of diet to our health. We're also going to see how our diets not only affect ourselves, but also possibly our children and our grandchildren. Now, as we continue to learn how Zoom works, we're going to keep adding features to make these talks more engaging. Last month, we introduced the interactive poll, and we're going to use that again here. Sanford Burnham Prebys discoveries have led to a very large number of patents. This means that our discoveries are being used out in the real world to improve the lives of people everywhere. Exactly how many patents have developed from our research? And here's the answer for you, which 21% of you got right. An astonishing 1,111 patents have been developed from Sanford Burnham Prebys research. So let's begin with our first speaker, Dr. Cynthia Lebopin. Cynthia is a postdoctoral fellow in the Degenerative Diseases Program at the Neuroscience and Aging Research Center at Sanford Burnham Prebys. Dr. Lebopin's career goal is to become a professor and research group leader investigating metabolic diseases. She obtained her PhD in 2018 in France in a collaborative lab of scientists and clinicians studying obesity. Her current research focuses on identifying the mechanisms responsible for fatty liver disease progression to cancer. Please welcome Dr. Cynthia Lebopin.
Thank you very much, Joe. It's my pleasure to be here. Okay, so today I'm going to present a little bit about my work here at Sanford Burnham Prebys, which focuses on high-fat diet and the development of chronic liver disease. So over 70% of Americans are overweight or obese, really making this a major public health challenge. And this can be caused by multiple things. It can be caused by biology, either a genetic predisposition or a hormonal imbalance, the environment, like the effect of endocrine disruptors, but in most cases is due to behavior, like an unhealthy diet or physical inactivity, which is the purpose of this talk today. So obviously, with our virtual environments, this isn't helping, but it's really important that we take care of ourselves because obesity can significantly affect all of the major organ systems within our body. It can increase the health risks associated with the nervous system, the respiratory system, cardiovascular system, digestive and skeletal systems, ultimately increasing the mortality of some of the leading causes of preventable death. And my research is focused mainly on exploring the effects of obesity in the digestive system, and notably the liver in the progression of these listed diseases. The majority of obesity-related liver disorders are called non-alcoholic fatty liver diseases, and it's affecting the majority, if not all, obese patients with no approved medication available and no non-invasive index, as it requires a surgical biopsy for diagnosis. So as the name NAFLD or non-alcoholic fatty liver disease implies, it's going to be characterized by the buildup of extra fat, the abnormal lipid accumulation within the liver, causing this liver to be yellow, enlarged, and basically look like a foie gras for humans. And as this disease progresses, it can arise to steatohepatitis, which is also called NASH, and is an evolved form of the disease showing inflammation, where there's going to be immune cells within the liver, potentially leading to cell injury and cell death. And that can lead to the appearance of scarring and fibrosis to try to kind of patch things up within the liver. This can then increase the risk of developing cirrhosis, where in this case you have increasing tissue damage and the appearance of regenerating nodules, which can then lead to hepatocellular carcinoma, which is the primary form of liver cancer and an aggressive malignancy. So the more this disease progresses, the less reversible it becomes, really making it urgent to catch it early and understand the dysfunctional mechanisms involved. So while HCC has mostly been characterized as an end-stage disease related to viral hepatitis, the emergence of antiviral therapies to attack this disease, and due to the exponential rise in obesity, has led to projections that liver cancer arising from NAFLD and NASH, as opposed to viral hepatitis, will significantly outnumber this cancer by at least 18-fold, coming from this NAFLD NASH instead of the viral hepatitis by 2025. And while cancer mortality trends within the US over the past 20 years have tended to decrease in the majority, it has significantly increased for liver and bile duct related cancers. And not only is the incidence of liver cancer increasing dangerously, but this liver cancer mortality has also doubled over the past 20 years. But what does your liver actually do? So your liver is your metabolic gatekeeper. It's going to assure essential functions, either helping with digestion, detoxification, modulating your blood sugar levels, and also recycling excess building blocks like from the macronutrients that you eat in order to be distributed throughout the body. So the cells performing these functions are called hepatocytes. And they each are going to contain many organelles here, like the endoplasmic reticulum, the mitochondria, and the nucleus. And sometimes it also accumulates lipid droplets, which aren't supposed to be there, but can create this lipid toxicity, increasing the ER stress, oxidative stress within the liver in the case of fatty liver disease. So what happens during this non-alcoholic fatty liver disease progression to liver cancer? Well, we believe that there is a vicious cycle of cellular stress occurring. Not only do you have this ER, oxidative, and lipid stress in the hepatocytes, it's going to promote the death of these hepatocytes, which can create an environment where there's going to be scarring and the appearance of fibrosis, inflammation, as I said, and the appearance of these immune cells to try to help and patch up what's going wrong. But also in the case of obesity, you're really going to have an involvement of surrounding tissue or organs, like for the intestine, where there can be an increase in gut permeability. And what this means is that you can actually have bacterial products leak within the liver, and also a leak from the adipose tissue of fatty acids that can increase the amount of lipid droplets that appear here, because excess fat is actually supposed to be stored in the adipose tissue, and now will come and lodge itself where it can, either in the liver, the heart, and that's how you get digestive and cardiovascular diseases. So our goal in the lab is to study the interplay between this nutritional excess, the metabolic reprogramming that can occur, and stress to the endoplasmic reticulum. This organelle is going to be involved in the manufacturing and packaging of proteins that can then be distributed throughout the body. And if you look at this really zoomed in image of a liver cell or a hepatocyte, you see a nucleus here, a small lipid droplet, and ultimately your mitochondria looks good, and your endoplasmic reticulum is the spaghetti-like form within the cell. In the case of fatty liver, this stress is going to become chronic. So our protein of interest is activating transcription factor 6, or ATF6. This protein is located on the membrane of the ER and is going to be responsible with helping the ER return to normal stages in the case of moderate or transient stress. However, in the case of obesity, the stress becomes chronic and the ER is not going to be able to come back to normal. It's going to be too much to handle for ATF6, causing a damaging ER stress response and basically turning ATF6 from a friend to a foe. If we were to look at what that looks like in terms of the liver, if we zoom in into the liver cell of a mouse that has eaten a high-fat diet or has fatty liver disease, we see that there's an accumulation of all of these massive lipid droplets that are kind of suffocating the liver cell. And within that, you have a stressed mitochondria, you have the ER, which is enlarged and swollen, indicative of a very strong ER stress response. So what we would like to test is whether ATF6 may be responsible for this high-fat diet and ER stress-driven liver cancer. So we use mice that are born either with or without this ATF6 protein, and in which we inject a liver carcinogen and place the mice on a high-fat diet to try to mimic this disease progression from healthy liver to NAFLD to liver cancer. And what we found was that in mice that still had ATF6, we had the appearance of this strong ER stress response, and the mice developed a significant number of these surface tumors on top of the liver, these very numerous and malignant lesions. And when we zoom into them, into this HCC nodule, we see that the tumor contains a significant amount of lipids and fat droplets that are accumulating. In mice that are born without ATF6, we found that that was sufficient to inhibit and prevent basically this disease progression to liver cancer. And when we look at this section here, we see that there is going to be a very small, potentially benign tumor, especially when compared to this mouse above, and no major accumulation of lipid or fat droplets. So our preliminary results lead us to believe that ATF6 can play a central role in fatty liver disease progression to cancer. So from there, our future directions are going to consist of studying how ATF6 exerts this cancerous role in the liver, whether different diets can modify ATF6 expression, and whether targeting ATF6 can be a therapeutic strategy. So basically, we aim to understand how diets really affect our body for translation from fundamental research here in mice to human applicability. And with that, I'd like to thank members of the Kaufman Lab, especially Randy Kaufman and Clementine Druel, our collaborators, SBP Core Resources, and the funding that makes this research possible. Thank you for your attention.
Cynthia, thank you so much. A couple of questions that I had in watching your presentation. One of which is, so this damage that's being done to your liver, we can't really see it until it has become a real problem, right? Is that correct?
Exactly. So the problem with this disease is that it's called a silent killer because in the early stages, there are no symptoms. The liver does a really great job in adapting itself and trying to do what it can in the early stages. And it is relatively reversible. So there's actually no ultimate exterior sign that we can see from the liver that is going to indicate a possible disease or disease progression. That's why this disease even requires a surgical biopsy. So actually going in and taking out a little piece of liver to be able to prove that it is indeed fatty liver disease.
And so really to protect ourselves against that, the best thing to do is to eat a sensible diet.
Yes, so sensible diet, exercise, all of these New Year's resolutions that you touched up on. And, you know, this fatty liver is reversible. So that's something great in the early stages. We find that there's a lot of elasticity within your liver, and it will take a while, but if you do follow up on diet and exercise in the long run, you can definitely reverse this disease and return to a normal liver until you reach to cirrhosis or HCC.
And in terms of what we eat in our diet, does it matter what type of fat? Is there a difference between saturated fats, unsaturated fats, trans fats, or do they all sort of contribute to this?
Yeah, so you know, exactly what you were saying. So there are technically good and bad fats, we can say. And you know, even the Food and Drug Administration recommends that we don't take in too many saturated fats. So a good, I guess, kind of comparison we could say is unsaturated fats, you can sort of recognize them because they're going to be liquid at room temperature, kind of a hint there. It's like your olive oil, your peanut oil, things like that. You also have some good omega-3s with you know, oily fish or nuts and certain veggies. And those relatively harmful fats are going to be the saturated fats, which are going to be solid at room temperature, like butter, cheese, things like that. And the trans fats, and that's going to be any kind of baked goods or fried foods.
Got it. Another question from our audience: how do statins help or hurt fat deposits in the liver and the liver generally?
So statins, for those who don't know, it's a kind of therapy that's used to regulate cholesterol content. And that's actually one of my therapeutic aims. I'm going to actually study the relative contribution or the potential therapeutic impact of statins on fatty liver disease. But for now, it's really going to be used for people with high cholesterol levels in reducing those cholesterol levels to a level that the liver can function and work with, because your liver is really going to be your body's filter. So any of that high fat diet, high cholesterol, it's going to be able to manage it, but only if you're in moderate levels. And that statin therapy has not been proved yet for fatty liver disease, but as it reduces cholesterol content, that is also one of the good things in therapeutic perspectives with fatty liver disease.
And then last question for now to follow up on that, in terms of pulling cholesterol out of the blood system, are there food choices that are good for doing that?
So again, I would recommend, speaking with a dietitian on recommendations for diet. But what I would say is to stick to elements that you can see, right? The less transformed, the better. So if you can't recognize the kind of food that you're eating, if it's been way too processed to understand if it's meat, vegetable, or what's in it, I would try to avoid that. And also junk food. So any kind of food that has been ultimately processed is going to be harmful. And just eating in moderation, 3 times a day, and to stay away from any high fat, high content, high sugar foods. But then again, not necessarily eliminating an entire food group from your regimen.
Okay, and that fits in. That was going to be my last question, but we have 3 questions about the keto diet. And because it's such a high fat content, and since the accumulation of fat causes the issues, can you talk a little bit about the relation, let's say, of the keto diet to what your research?
Yes. So the keto diet, again, is potentially harmful for the liver because it does indeed rely on high fat content. And it also doesn't necessarily discriminate between good and bad fat. As we were just saying, this keto diet allows saturated and unsaturated fats. And it's not going to be a relative good diet to use just because you're going to be providing fat from the diet that the liver can then store itself or distribute elsewhere. So although there's a big hype around these diets, it's also not been studied in the very long term. So there are a lot of short-term effects that come when you eliminate food groups from a diet, but there's going to be too much risk with adding too much fat, and also risking nutrient deficiency. So that's the side effect that everyone should be aware of. If you do eliminate entire food groups or rely on only one type of fuel, like the keto diet for increasing the fat content, if you don't eat a wide variety, you might be at risk for deficiencies in vitamins and minerals. So not necessarily something that I'd advocate for either.
Got it. Thank you, Cynthia, very much. We have other questions here, but we're going to move on with our speakers, and we'll try to get to those questions at the end. All right, our next speaker is Dr. Erin Havas. Erin is a postdoctoral associate in the Tumor Initiation and Maintenance Program in the NCI Designated Cancer Center at Sanford Burnham Prebys. Dr. Havas studies the epigenetic mechanisms of aging and how they relate to disease progression in cancer development. His research includes how aging mechanisms promote non-alcoholic fatty liver disease and cancer development, and how diet can affect those mechanisms.
Hi, everyone. So what I'd like to talk about is a rather similar question to what Cynthia was addressing, but instead, for me, I'm interested in a combination between, what's the effect of aging and diet on liver disease? And ultimately, how does this lead to increased risk of developing liver cancer? There are roughly about 35,000 cases currently being diagnosed every year of liver cancer in the United States, which puts it at relatively a minority of the cancer. It's not in the top 10, but it's a really dangerous disease. It has a 5-year survival rate of about 18%, which means only 18% of those people that are diagnosed with liver cancer likely make it to the 5-year point. And this shows itself in the estimated cancer deaths. Liver cancer is roughly, it's one of the more deadly cancers, and it constitutes in males the top number 5 cancer for cancer deaths, and number 7 in females. And one of the things that's happened over the past 20, 30 years is that we've seen an increase in the incidence of liver cancers being diagnosed. So here we can see in 1999, we had about 12,000 some odd cases that were diagnosed, but in 2016, we were about 33. So this rate is more than doubled over 20 years, and that seems to be, progressing continuously. We want to ask the question, why is liver cancer incidences increasing? We do know the top risk factors for developing liver cancer, which are things like obesity and aging. Males are more prevalent than females. Viral hepatitis, like Cynthia says, has been better treated, so it's less prevalent in what's driving it. But diabetes is also on the rise, and excessive alcohol consumption. And for this talk, we're interested in a few of these. The top one right now is obesity. And one of the things that we have to address is that America really is getting fatter. We can see here, this is from Forbes, this graph, but the red group, we can see in 1999, roughly about 30% of the population was considered to be obese. Now, however, that population is almost 40%. So we've seen this increase over the same period of time that we've seen an increase of liver cancer take place. And like Cynthia said, roughly about 70% of the population would be considered to be either obese or overweight. And that's very, very dangerous. Like Cynthia had shown, the same graph, right? We see obesity that promotes the non-alcoholic fatty liver disease, which includes all of these steps, including the fatty liver, the hepatic steatosis, the inflamed liver, which is non-alcoholic steatohepatitis, ultimately leading to deposition of more fibrosis, scar tissue coming into the liver, and seeing repopulation within the liver of nodules. This is cirrhosis that can lead to hepatocellular carcinoma. But one of the reasons why I really like to show this graph is it kind of shows an underlying thing, which is that this is not a progress that takes place overnight. You don't become fat and then you just have a fatty liver. Instead, this is something that's progressive and takes place over many, many decades, 20, 30, 40 years even. And a lot of it is reversible up to a point. And so, because this is something that takes place over decades, we have to take age into account. And that's really where my research focuses on, is what is the, age-dependent aspects of the fatty liver disease development and cancer, right? So we have obesity and aging. And the reason why I point out aging specifically is this. These are the number of new cases diagnosed per year, broken down into age groups, right? So we can see that, for an individual that is, say, 20 or 30 years old, the risk of developing liver cancer or being diagnosed with it is rather low. But by the time you reach about 55 years and older, now that rate goes through the roof. And this is pretty true for most cancers, that in reality, these cancers are an age-dependent disease. And a lot of models don't really take that into account, but it's very, very important. The reason is that there's currently a higher percent of population that's now older. So this is from the 2016 census. And what we can see is that, that we're having an upwards trend of the population being older, right? By the time 2034 rolls around, we're going to be seeing a shift of there being more adults age 65 or older as compared to children that are 18 or older. And this has been termed as the silver tsunami, right? So Graham Mackey has this wonderful, depiction here where we can see that there's a lot more senior citizens coming through. And this really lays down some rather important questions that we need to address in health, which is, maybe we can have these individuals age in a more healthy way in order to hopefully prevent some of these age-related diseases like cancer. And so when we ask the question, ultimately cancer is an age-related disease that's associated with an accumulation of DNA damage. And this is true for most cancers if we forget about pediatric cancers. And these DNA damaging agents, like for instance, we know about UV light from the sun, chemicals in the environment, ionizing radiation. These are all environmental aspects, but there's also internal aspects as well. So metabolism that release reactive oxygen species can cause DNA damage, and also DNA replication errors. Given the rate of errors that we have, by the time we're about 6 years old, we've seen, our bodies have probably seen a cell that could become cancer. But generally, these are eliminated by the immune system. Our immune system's really great at eliminating those cells. And that's why we don't normally develop cancer until we're in our 60s or 70s. But when we can't necessarily eliminate those cells, what happens is that, the cancer comes from us. It is our own cells that have mutated. And if they're not eliminated, then what happens is they start to, multiply. Then these blue cells are now turning into a larger group. And then over time, they can become a malignant cancer and can metastasize to different regions in the body. And so what we normally see is we have here, we'll say this is a liver tissue in brown, but we have this abnormal cell that's appeared here in blue with the spikes. And so what happens is under normal, young, healthy conditions and tissues, this causes immune signaling to take place. So pro-inflammatory signals come out, and this causes recruitment of immune cells. As we can see in the green here, these immune cells then become activated. They target the cancer cell for elimination. That way we can get rid of the cancer cell, and eventually we can repopulate the area and have a nice healthy tissue again. But we believe that this process changes a bit with age. We want to understand that, these events more in age. So when we ask the question, what are the effects of age and obesity in the liver? Because we do know that obesity and age come together in order to create and ultimately something that leads to more risk of developing cancer. So what I did is I take young mice aged about 3 to 6 months old, so they are adult mice, and I take old mice, which by about 18 months of age, that's roughly a geriatric mouse. And then what I do is I provide a very similar diet that Cynthia does to her mice, but I only give it for 9 weeks. And so for those 9 weeks, they receive 60% of their calories from fat. So imagine if you take all of your food, but you just kind of coat it in mayonnaise for this whole time. It's a pretty extreme diet. Not everybody necessarily can get that much fat in their diet, but mice are in a controlled environment, so we can test this. And what we see is after that 9-week period, we now have obese mice. And so what I did is I look at the gene expression changes in these mice. So what we see here, this is a comparison of genes. So genes can be either turned on or turned off, given whatever stress or to their environment. So our basal level in young mice, we can see the genes that are turned off or lower expressed. These are blue, and we can see each one of these bars is a gene of interest within just a subset of genes we are interested in. And what we can see is, so a young mouse has a lot of blue, right? A lot of the genes are either downregulated or turned off. And when we give the 9 weeks of high fat diet, we do see some genes that appear in red. And the same is true for just age. So old, normal, healthy mice, we do have some more red. So there is a little bit more gene expression in this group. But when we give high fat diet to an old mouse, the gene signature changes dramatically. We see a whole lot more genes going up with the high fat diet treatment as compared to just the young mice given the same exact diet and time. And a lot of the genes that are in this group are associated with not only just metabolic functions, but also with inflammation. So pro-inflammatory regulation and also immune regulation is what we're finding out. And we believe that this group here really holds the key to understanding why it is that maybe age and diet can come together in order to create an environment that allows for development of cancer. So my model that I have currently is we have a tissue, a liver tissue here, and we do have maybe some abnormal cells, but we now have had long periods of pro-inflammatory signaling coming out, which means that we can, the immune cells are recruited, and generally we see a lot more immune cells in old mice than we do in young mice. But what happens is when we're giving the high fat diet, now we start to express these immune modifying proteins, right? These little stars. And what these stars do is they prevent the immune system from being able to become fully activated, and in some cases can actually turn off the immune system. And what this does is this allows for those cancer cells to begin to populate and not be regulated by the immune system. So for me, I'm trying to understand, are there ways that I can target these functions, the gene regulations in hepatocytes in the liver, in order to ultimately prevent the aging effects and possibly be able to reverse some of the aging effects and things that are associated with development of liver disease and ultimately liver cancer. And so with that, I would like to say thank you. I really want to highlight my mentor, who's been wonderful to me for the past 4 years, Dr. Peter Adams, and then my wonderful lab, who has been extremely supportive, along with the funding that allows for this research to take place. And with that, I would love to take any questions.
Aaron, thanks so much. It sounds like to me what you're saying is that as we age, our diet becomes even more important. That what you're seeing with older high-fat diet mice. So are we saying? I know we're not saying definitively, but are you seeing that what we need is that, or that it would be smart that as we age to reduce the amount of fat in our diets in general?
I'm not sure if I should say that we should reduce the amount of fat. One, by giving 60%, this is a really extreme amount of fat that we're given, right? But I think that as we get older, we must absolutely pay more attention to what we eat and ultimately how we use what we eat, right? Exercise becomes more and more critical. And my data is showing that really with age, responses are just different. They can be more extreme in an old mouse given the same exact condition.
Got it. We had a question about liver transplant immunosuppression drugs. And we are talking, do those cause or exacerbate problems with the processes you've talked about?
Well, so by the point that you have a liver transplant, which I believe it's called the Whipple procedure, you are now taking a bit of hopefully healthy liver from another individual, and you're giving it to somebody who ultimately needs a new liver, right? So at that instance, when we're giving immune suppressants, the idea is so that way the immune system, your immune system, doesn't target the new tissue that's put in, and also the immune system that kind of came with the tissue doesn't target you as well, avoiding graft versus host. So it's a bit different as compared to what I'm looking at. I'm looking at more along the lines of what's the signaling events that are taking place under normal conditions. But immune suppression is really important for surviving a hopefully curative treatment like a liver transplant.
But it sounds like also in some of your research that one of the things that's happening is the immune system is being suppressed. So as we age, if we continue to eat unhealthily, we are in some ways suppressing our immune system, not to the extent, let's say, that immunosuppression drugs, but that is an effect of what we're having. Is that a correct interpretation?
Yeah, I mean, the immune system is definitely able to be affected by things like exercise and diet. There's been studies that have been shown, for instance, as we get older, we can't reproduce more and more of these T cells that are really important for responding to things like cancer, because we have fat that builds up in our thymus, which is important for T cell development. But there's some really interesting studies that older people that have exercised for long periods of time, meaning years and years, that they're actually able to help kind of clear out some of the fat from their thymus and be able to respond. So the immune system does change in that way. And also within my data, where you can see that the immune system within the liver itself alone does in fact change.
Got it. Last question, and this you may be the right person. We may need to bring it back at the end. Are there common medications that can cause or affect some of these liver problems in addition to diet that you're aware of?
That's a really good question. The answer is likely yes. I have been exploring some effects of chemotherapy drugs and being able to increase the rate of aging, for instance. So we do see in a young mouse, whenever I give a drug that causes DNA damage, I can kind of speed up the rate at which that mouse is now aged in the liver, and show some of similar responses, increased inflammation that we see in an old mouse as well. So the answer is probably yes. Things like Tylenol toxicity can affect the liver as well. I haven't explored that in massive depth though.
Aaron, thank you very much. This was very good. We're going to move on to our last speaker now. We are also going to put up our last poll question. Which of these foods has the fewest grams of fat? And while you're noting your answer, I'd like to introduce our last speaker, Dr. Karen Okor. Dr. Okor is an assistant professor in the Development, Aging, and Regeneration program in the Neuroscience and Aging Research Center at Sanford Burnham Prebys. She uses the fruit fly to investigate genes that contribute to heart disease, and she has pioneered methodologies that allow her to examine the cardiac effects of genetic manipulations in this model organism, which shares surprising similarities with human hearts. Her presentation will illustrate the effects that high-fat diets can have on our hearts and why that is so dangerous. And speaking of high fat, let's look at the results of our poll. And 12% of you got it right. Surprisingly, the donut has the lowest fat content. I'm going to take that to mean that donuts are healthy, though I'm sure Dr. Okor will disagree with me. Please welcome Dr. Karen Okor.
Thank you very much. It's a pleasure to be here. And I'm going to switch organ systems from liver to heart. So despite years of research, both in clinics and in labs, heart disease is still the leading cause of death in the industrialized world. And you might wonder why I chose to use fruit fly to study that. And hopefully I'll convince you by the end of my presentation that it's not a bad idea. But the first reason is that, or the biggest reason, I guess, is that the causes of heart disease are very difficult to determine because there are so many different things that can influence heart disease itself. So one of the main factors, of course, is obesity. And so in addition to causing liver disease, it also causes heart disease. If we look at the incidence of obesity in the United States, with the darker colors representing greater incidences of obesity, and if we look at this same map with the rate of hospitalizations for dysrhythmia, you can see pretty clearly that there's a direct overlap between the higher rates of obesity and the higher rates of hospitalization. And I want to point out that one of the main dysrhythmia causes or problems is atrial fibrillation. And I'll talk a little bit more about that later in my presentation. So in addition to obesity, age also is a factor in arrhythmias. And you can see here that in general, the higher incidence of high age overlays with the higher incidence of dysrhythmia. So in addition to both obesity and age, genetics also plays a big role in the incidence of heart disease. And so in the case of atrial fibrillation, there have been a number of studies looking at human genetic analyses to identify genes that are associated with AFib. And about 200 genes have been described. And if we try to see how these genes might interact together, we end up with this big hairball from drawing from information that's already known about these genes. And so it's very difficult, especially in humans, to try and figure out what the interactions are between all of these genes. And then to overlay diet on that, as well as age, it becomes something that's nearly impossible. So because of that, we've turned to a simpler system, the fruit fly, in order to help us try to figure out what's going on. So fruit flies do have a heart that's pretty important, and it's a heart that functions like your hearts. It beats on its own. It doesn't need any neural input. Importantly, fruit flies have about 75% of the genes that cause diseases in humans, and that includes the cardiac diseases. It has a short lifespan, so that allows us to look at the interactions of aging with heart function in this model system. And just so you know, a fruit fly that's 1 week old, it's about equivalent to 10 years in a human lifespan. So within a couple months, which is the full lifespan of the fly, we have gone from a young newborn to an equivalent of an 80-year-old human. And we have loads of genetic tools. This animal has been used for 100 years to try and understand genetics. And they're small, which is nice because then we can keep a whole lot of them in our labs. We keep them in vials that look like this. And for relative size, that's about the the circumference of that vial is about the same as a quarter. And we have figured out how to study the heart, which you're seeing here. It's an abdominal heart. It's linear, but that's the same in your heart when you're developing in utero. Your heart starts out as a linear tube. And we can take high-speed movies of these hearts beating, and we can get a lot of information just from these movies. We can do something with them, which is similar to what your doctors do when you have an echocardiogram, and that is take a slice out of this movie and electronically line it up, every frame, that same slice from every frame in the movie, so that you can see in one shot, the movement of the heart tubes during a contraction and a relaxation. And we get a lot of information from this. So I already showed you how the incidence of cardiac arrhythmia increases with age. Well, it turns out that's also true in flies. So this shows you that, that picture called an M mode from a young fly that I just showed you. By the time they're 5 weeks old, or the equivalent of a 50-year-old human, you can see that there's some clear arrhythmias that are showing up. So this gave us a lot of encouragement that we could use this fly to understand what's going on, both as we age and with diet. So we feed normally we feed flies a mixture of yeast and molasses, and sometimes we add some sugar. But if we feed them with that same diet supplemented with 30% coconut oil, after only 5 days, you can really see the effect on this heart. It's a hugely lipotoxic effect that is also shown in this M mode here. Now, what is 30% fat in your diet? Well, it turns out it's not a lot, but it is what is recommended by the USDA. So they recommend 20 to 35% of daily calories should be fat, not more. And if you weigh 140 pounds, that's roughly 70 grams of fat each day. What does that look like? Well, this is about 70 grams of fat. And you can see that it's not going to take a lot of fatty foods to get you to that limit. And so that's why keeping track of how much fat you eat and limiting that is going to be helpful, not just for liver, but also for heart. We also found, using this high-fat diet system, that the effects of a high-fat diet can be transmitted from parents to offspring. So if we take the flies that are fed this high-fat diet, as well as flies that are fed the normal food, and we let them lay eggs and look at heart function in their children, we see that the children fed on a normal food diet that came from flies fed a high-fat diet, those children also have heart dysfunction, as suggested here by this broken heart. Now, this is something that also happens in humans. It's just we can't study it rigorously because it would be unethical to make people eat a given diet. But it has been done indirectly, for example, during the war when there was a blockade on the Netherlands and there was mass starvation in the country. The children that were born during that period were much smaller than normal. They had a tendency to be diabetic when they got older, as well as to be more obese and have cardiovascular diseases. And so we know this happens in humans, and we're using this model system to try to understand that. In fact, it takes 2 generations in the fly anyway to get rid of this parental effect of a high-fat diet on the children. So we also know that genes cause cardiac defects in flies just like in humans. And if this is a normal heart, here's a mutant heart. And this mutation is in one that is known to cause athletes to have sudden cardiac arrest when they're playing a basketball game, for example. That same mutation in the fly causes quite a large amount of cardiac arrhythmia, as shown here in this M mode. So what we're doing is taking this really nice model system with lots of good genetics, and we're trying to combine it in a cross-platform collaboration with other labs here at SVP to try to identify, of those 200 genes that I showed you, the atrial fibrillation genes, to try and identify which ones actually are responsible and how they might interact to cause atrial fibrillation. So we have developed a high throughput system where we don't have to dissect open the fly, and we can just take a picture of its heart function from through the cuticle. And we're combining it with experiments, high throughput experiments in human-induced cardiomyocytes. And what we're doing is testing all of those AFib-related genes that form that big hairball. And the ultimate goal is to identify genetic networks that are involved in AFib. So we have started this, and here you can see that big hairball I showed you earlier. We've tested quite a number of genes already that are circled in red to try to understand which ones cause cardiac arrhythmias, both in flies as well as in the human-induced cardiomyocytes. And the ones that we found that cause arrhythmia in both systems, in both cell culture as well as in an intact organ, are circled in yellow. And so you can see we're starting to home in on some of the key genes that we might be able to put together to explain what goes on, at least in one form of arrhythmia called atrial fibrillation. And so far, this is the network that we've identified. Here are some of those yellow circled genes shown here. And we've picked out a really interesting subnetwork already. And that subnetwork is interesting because it contains a gene called DSAT. And DSAT is an enzyme that produces monounsaturated fats, or MUFAs, from saturated fats. So it's thought to be potentially protective, that particular gene, for heart arrhythmias. But we now, whoops, did not mean to do that. We now have a network that contains not only that enzyme, but also a number of other atrial fibrillation related genes that are shown both in yellow and orange here. And we are going to be further trying to understand in the fly the interactions between those genes in order to develop testable and treatable networks underlying fibrillation and arrhythmia in the fly heart. And with that, I want to thank the labs involved. So my lab, as well as Rolf Bodmer's lab, are working on the fly aspects of these network testing platforms. And the Alex Kola lab is pioneering the human-induced pluripotent stem cell cardiomyocytes. And also thank our funding. And thank you for your attention.
Karen, thank you very much. So similar to the question that I had asked Cynthia, in regards to specific diets like a keto diet or a paleo diet, which are specifically high fat, are they unsafe for the heart? Is that something that you're finding with your research?
Absolutely. Any high fat diet is unsafe for the heart. It's quite toxic. The heart can't really and wasn't designed to store fat. So it has to bring in fat from the blood to fuel its very active beating patterns. And so if you have too much fat in your blood, the heart's designed to pull it in, but it doesn't have anywhere to be stored, and it begins to cause problems with the cell membranes, as well as just overall causing problems in the arteries that are feeding the heart. So blocking blood flow, which provides the oxygen as well as the nutrients that the heart needs. So it's a multifaceted problem if you have a relatively high fat diet.
And another question here, and then we're going to bring Cynthia and Aaron back on and ask questions, but what is considered obese? I think we talk about obesity and a lot of people don't necessarily know what that means. How many pounds overweight, or is it a body mass index?
Yeah, it's the body mass index. If you have a body mass index over 30, that's considered obese. So that's a function of your height and your overall weight, essentially. But I believe the latest guidelines have that if your BMI is over 30, you're obese.
Got it. And we're going to ask Aaron and Cynthia to come back on. There was a question about the reversibility of heart damage and calcium and plaque buildup, for example.
Unlike the liver, the heart cannot regenerate. We are trying to figure out ways to make it regenerate, but we haven't gotten anywhere close yet. But the problem with heart disease is, and especially in the case of a myocardial infarction, when you damage the cells, there are no stem cells, there is no way for new cells to be made to fill in that damage. So what happens is you get fibrosis, so similar to damage in the liver. The heart makes a fibrous network to fill in that damaged spot, but that doesn't go away. And in fact, it tends to increase with time, even though it's not needed anymore. And that part of the heart is no longer contractile then. So that damage is permanent.
Wow. Here's a question for everybody. And just to sort of play traffic cop here for a second, we'll start with Cynthia. What about high sugar and other high calorie diets, or alcohol or fructose? What about other things other than fat and the effects that they have that you're seeing in your research? So Cynthia, let's start with you.
That's definitely a great point. So you don't necessarily have to eat fat to have a fatty liver. The liver itself is actually capable of creating fat to store it from any macronutrient source that includes fat, carbohydrates, and protein. So it's also been shown that feeding mice a high fructose diet is going to induce the similar effects of fatty liver disease and the same risk to liver cancer as high fat. And in terms of how that applies a bit to humans today is there's good fat, there's bad fat, there's also good sugar and bad sugar. And the one sugar, that always comes up is the high fructose corn syrup sugar. So any of that processed sugar that's not natural is going to be harmful as much as fat, if not more.
Aaron, anything to add there?
Yes, I do actually.
Oh,
So
Karen.
I
said Aaron, but that's okay. Go ahead, Karen.
Oh, sorry about that. We've fed high fructose sugars to our flies, and we do get heart damage, but it's a different kind of heart damage than we get when we feed them high fat. So the heart can't really do anything with the nutrients it brings in, but if you feed a fly a lot of sugar, they become fibrotic, more so than that arrhythmia kind of phenotype that I was showing.
Got it. And now, Aaron, we'll go back to you real quick. Do you have anything to add to?
I mean, I think the question really is asking about what's termed is the Western diet, right? When you have a high level of sugar along with high level of fat, or at least a moderate level of fat. And Cynthia is 100% correct. Really, it boils down to very high risks of developing not only a fatty liver, but also it's associated with a lot of other different cancers that aren't necessarily directly connected to the gastrointestinal. It can affect things like breast cancer risk and stuff like that too. So the Western diet is rather dangerous, but like Cynthia said, those sugars ultimately can be turned into fats and can also lead to, higher levels of insulin resistance and progression of disease development.
So unfortunately for me, even though the donut was the lowest fat content of all those foods, it's not any healthier for me than anything else. All right. The source of the fat was a question here we had. If you're getting fat from nuts or seeds versus meat, is 10 grams of fat from meat equivalent to 10 grams of fat from nuts? And Karen, you're already shaking your head, so I'm going to let you jump in first, and then we'll go to the others.
Well, I think it's generally agreed that monounsaturated fats are better for you than saturated fats, but it turns out that even monounsaturated fats are different depending on their source. So if they come from animals, it turns out that you will have a higher incidence of heart disease anyway, compared to people who are eating the same monounsaturated fatty acids from a plant source. So if you get your fat from plants, that's a good thing. If you get it from animals, it's probably bad, regardless of the kind of fat you're getting.
Got it. Aaron?
Yep. So a lot of the differences between the types of fats you take in, say from a plant source, as compared to an animal source, is going to be like Karen said. Are they branched? Are they saturated? Are they, and also one of the things associated with animal fats is we usually cook animal fats, which means we get higher levels of trans fats. And those are really the baddies, right? Those are the ones that have massive pro-inflammatory effects and can promote disease as well.
And Cynthia, anything you'd like to add to that?
Just a final point saying that, in addition to what Karen and Aaron have said, that eating it from plants can also increase your fiber content as well. And so fiber has also been shown to improve digestion and to kind of help the bad fat kind of go through the transit of your digestive tract. So ultimately, there is definitely less risks with the vegetarian kind of fat than the animal meat fat. Less red meat, basically, more lean meat if you have to.
And more donuts. No more donuts. All right. Thank you, Cynthia, Aaron, and Karen, and thank all of you for joining us. We have some questions that weren't answered, which we will put to our scientists and see if we can get some answers for you. If you have other questions, you are welcome to send those along to my email too. If you would like to learn more about or contribute to any of our research, you can also use that email. And then we'd like you to join us for our upcoming events. On February 23rd, we will talk more about heart disease and how to mend a broken heart. Perfect for Valentine's Day. And then on March 23rd, we will have sort of a one-year reassessment of where we are with COVID-19 and what the path forward is. So we invite you to join us for those. If you register for this, you will receive invitations to those presentations as well. Thank you again for attending today. Feel free to share the recording with your friends and colleagues. Good luck with those resolutions, and here's to an exciting 2021. Thank you again.